Hello, and welcome everyone. In this podcast, we're going to talk about gene regulation as it occurs in eukaryotic cells. The regulation of genes in eukaryotes, we'll find, is a little bit more complicated than we saw in bacteria. There are more, let's say, obstacles that have to be overcome in order to get the right genes turned on at the right time of development and in the right cells. So let's write gene regulation up here in eukaryotes. And it's important to remember, we've said this before, but it's worth saying again, that you may have a pancreatic cell here, you may have a neuron cell here, with the long axon here. Each of these cells contains a nucleus. Each of these cells contains the same 23,000 genes that are present in humans. And we could talk about other organisms as well. I'm just going to refer to humans here. Even though they have the same 22 or so thousand genes, the pancreatic cell doesn't want to express, so I'll put a minus, neurotransmitters. However, the neuron does want to express, I'll abbreviate this because of space, express neurotransmitters. However, the pancreatic cell does want to express insulin, but the neuron doesn't. It's important to realize a few things here as we get started. Genes have to be regulated in a number of situations, and they're regulated differentially in these different situations. And these situations include cell type or tissue type, which cell, which tissue are those cells in, as we see up here. We also have to think about development, particularly when we're thinking about a multicellular organism. Yeast are a single-celled organism, and so their level of development is not as great as, say, a multicellular organism like a fly, a worm, or a human. And another thing we'll talk about, particularly towards the last in the last podcast, is sometimes it matters the origin of the gene. That is, does it come from the sperm or egg? It seems a little hard to believe, but if you get some mutations from your mom, it gives you no phenotype, no adverse effects. But if you get it from your dad, it does give you an adverse effect. The flip is also true. Some genes, if you get them from your mom, it gives you an adverse effect. If you get them from your dad, it doesn't. So all this kind of makes eukaryotic gene expression a little bit more complicated than we saw with bacteria. So let's talk about some general ways that eukaryotes can regulate their gene expressions. So let's draw a big eukaryotic cell here. Remember, in eukaryotes we have a nucleus, so let's draw a big nucleus here. And over here we'll have our cytoplasm, of course. Remember, in this nucleus we have our DNA, and this DNA is transcribed just like we saw with in bacteria, and we get our messenger RNA. Then this messenger RNA, before it is released into the cytoplasm, it has to be processed into this mature messenger RNA, and then this mature messenger RNA leaves the nucleus into the cytoplasm. And I'm just going to put mRNA, and I'm going to put a little star by it just to indicate that it's been processed. It is then translated into a protein, and just like we saw with the bacteria, there can be post-translational modifications. So much of this looks like what we would see in bacteria. we see regulation at the level of transcription. However, remember, the DNA in a eukaryotic cell is much more complex. It's packaged up into nucleosomes that have to be removed in order to transcribe a gene. We have regions of the chromosomes that are heterochromatin, which are rarely transcribed, and then we have regions that are euchromatin that are often transcribed. A lot of things have to happen to this DNA before transcription can even occur. And then when transcription occurs, we still have those same kinds of regulatory elements. We have the promoter that needs to be turned on. We have some upstream elements which are new to eukaryotes, where enhancer and silencers sequence exist. And then this is a new area here in eukaryotes where that messenger RNA, before it can be sent to the cytoplasm to be translated, it needs to be processed. Remember, those introns have to be removed. It has to be capped with the modified G guanine, and it has to have that poly A tail added to the 3' end. Once it's in the cytoplasm, it'll be translated into a protein. There are some regulatory actions that occur at translation in bacteria, but we see a lot more in eukaryotes. For instance, in the cytoplasm, this messenger RNA can be silenced using special kinds of RNA molecules called microRNAs or short interfering RNAs. And we'll talk a little bit about those towards the end of these podcasts. Then once the protein is made, there's a lot of post-translational modifications that occur. We didn't think this happened too much in bacteria before, but we're starting to see more and more examples of it. But clearly in eukaryotes, this is a major factor in regulating whether or not a protein is active. And I should have ended here with our functional protein. Over these next few podcasts, we're going to talk a lot about the role at transcription, in part because we know probably a little bit more here. So we're going to talk about a few things over these next several podcasts at transcription here. We're going to talk about histones and nucleosomes. I'll just write histones here. How they are removed or added to affect gene transcription. We're going to talk about the elaborate network of transcription factors eukaryotes have. We're going to talk about how the DNA can be modified to affect transcription, and we're mainly going to talk about methylation, but there are a few other things that happen here too. So we'll learn that when genes are methylated, or areas of those genes are methylated, that can reduce transcription. And we'll learn something that is within the field of genetics, this is my all-time favorite topic, and that is how this methylation within a gene can be passed on to the next generation. So not a mutation, but whether or not the gene is expressed by these methylation factors. And that is something called epigenetics. Epigenetics has been around for a few decades, but it is still considered a relatively new area. We're starting to learn so much more about epigenetics. In many ways, it's changing our entire view of genetics. But within epigenetics, we'll talk about how this affects gene expression, how this can lead to certain kinds of diseases, how this affects whether or not the X chromosome is expressed. We'll even talk about an example with honeybees and how honeybees use the environment to control gene expression and ultimately what kind of bees are produced. This area of epigenetics also gets back to the area that we had on the last slide where we talked about the fact that it matters which gamete you get a mutation from. It matters if you get that mutation from your mother or from your father, and we'll talk a little bit about that too. It's a really cool area. And the other level that we're going to spend some time on is here, where we talk about these microRNAs, how they affect transcription, and these short interfering RNAs and how they affect. And then lastly, we'll talk about this processing here. Don't really have room, but we're going to focus here on alternative splicing. So that gives you a general idea of where we're going to go with the next few podcasts to explain gene regulation in eukaryotes. In the next podcast, so this is a pretty short podcast, just kind of opening the scenario. In the next podcast, we're going to focus on histones and transcription factors. Then the next podcast, it'll be methylation and epigenetics. And then the fourth podcast will be alternative splicing and microRNAs and these short interfering RNAs. Well, that's it for this introductory podcast for eukaryotic gene regulation. If you have any questions, let me know.